really live somewhere.
So,
yes, I think now we are live in YouTube.
Okay, good.
Yeah, great. So, good afternoon, everybody here in Mexico or in the United States or wherever in the world you are following this YouTube video. Actually, you can comment there now live. Um, Today we have our monthly seminary from the EMBS, the Society of Biomedicine of the IEEE, Puebla chapter, Puebla in Mexico. And today is a pleasure to have a friend here with us. He has been here in Mexico several times, and we have passed very good moments sharing some science here and talking about porous silicon. Here, let me introduce our speaker is Professor Mike Saylor. That I told you is a good friend of us, of the Mexican community. I will give you some points about his resume. I would say he was trained as a chemist, and actually he's now a professor, a distinguished professor in UCSD in the University of California, San Diego. He has supervised more than 160 undergraduate, graduate, and postdoctoral students of the UCSD. He has published more than 300 peer-reviewed papers, 7 book chapters, 1 book, and he holds 29 US patents and 6 international patents. He was named a highly cited researcher by Clarivate Analytics in 2018 and in 2019. He has a citation age index of 91. Actually, it's very, very, very high. Saylor serves on the editorial boards of the journals Nanoscale Horizons, ACS Nano, Applied Physics Letters, and Advanced Materials, and he is an associate editor of the journal ACS Sensors. He has served on several scientific advisory boards, including the United States Air Force, Illumina, Matrix Industries, Nanovision, Biosciences, Pacific Integrated Energy, True Tags, and Well Healthcare Technologies, and he co-founded Spinnaker Biosciences and Impilo Therapeutics. Saylor is an elected fellow of the American Association for the Advancement of Science, the Royal Society of Chemistry, and the National Academy of Inventors. Yeah, it's a great pleasure to have you here. I'm tired of reading your resume.
It's fine. Thank you, Enrique. It's fine. It's a great pleasure to be here and great pleasure to talk to you and see you again and meet some new friends as well.
Yes.
Let me go ahead. Are ready? You ready for me?
Yeah, you're ready.
Okay.
Yeah, you can.
Okay, I'm going to go onto my full screen here. Let's see if it works.
Yes.
Okay.
Yeah, it works. Yeah, you can start.
One thing that the university asked us all to do is to cite some of our potential conflicts of interest, and so that's what this slide is for. I think, Enrique, you actually mentioned some of these companies, a few companies we work with to translate some of the technologies you hear about today in the talk, and some other companies that are not related at all to what I'm talking about, but just as a full disclosure. So, I want to start this story, since this is a biomedical community, it's pretty easy to start this story with a bunch of images that you guys are all familiar with, I'm sure. This is a picture from the website for the drug Abraxane. Abraxane is an anti-cancer drug. Actually, it's one of the successful nanotechnology drugs or nanomedicine. So they don't call it a nanotherapeutic or a nanomedicine on their website, but if you look deep enough in the web page for Abraxane, as is true for many nano-based medicines, you'll find some pictures, something like this, where they show what is essentially a nanoparticle. In the case of Abraxane, it's an albumin colloid, basically, a nanoparticle made out of albumin with paclitaxel as the therapeutic. Paclitaxel is a small molecule anti-cancer drug, and it was found that by loading this very toxic drug into a larger nanoparticle, you could make the drug more effective. So I'm going to talk a lot about that concept of putting a drug that may not be so effective on its own into a larger structure. And where I want to kind of pivot to talk about how we think about these nanoparticles kind of like robots. And so you start out with the concept of a robot, which is developed in the early part of the 1900s. Isaac Asimov, who is a science fiction author, wrote this book called I, Robot. They made a movie out of it recently with Will Smith. I think maybe some of you saw that. Anyway, within this book, Isaac Asimov pointed out that robots should have certain laws or rules that they follow, and he called them the laws of robotics. And the three are shown here on the left. So robot should not injure a human being or through an action cause a human being to come to harm. The second law says a robot must obey the orders given it by human beings, so it must follow its programming. And then third, a robot must protect its existence, so it shouldn't just jump off a cliff if it doesn't have to. So protect its own existence unless it's conflicting with its other laws, meaning to protect itself, it has to kill its a human. That would be against the first law. So that's what those laws were. So many years ago, I decided to think about how you would take these nanoparticles we talk about as nanotherapeutics and think about them in terms of robots, and really more in terms of calling them then nanorobots. And those types of robots also have similar rules, I think, that we all should be following when we design these structures. The first one, basically following on Isaac Asimov's law, is that the robot shouldn't harm humans. And so in the case of a therapeutic or a nanoparticle that's going to be injected into the body, basically that means it shouldn't be toxic or otherwise harmful to the patient. The second law, which really says that it has to follow its programming, is true also in the nano world. But the difference is that in nanoparticles, we can't write lines of code to identify a certain function or certain process by programming. So all of our programming, all of our lines of code that we write in nanorobots is done using chemistry at the very small, basically molecular level. And so just think about the chemistry that I'm going to talk about today as being the lines of code that we're writing into our computer program that tells our robot what to do. And finally, the third law, which Isaac Asimov said the robots must protect themselves, the nano robots, I think, have to conflict with that law. They should not protect themselves at all costs, or at least once they're done doing what they're supposed to do, they should self-destruct. Because when you're putting something into the body, you don't want it to last much longer than it has to last. And so this is a little bit different than Isaac Asimov's rule, which the robots would be around forever helping people, plugging themselves in and charging themselves up every night and then continuing to work. Once you've cured a disease or treated the patient, you really want that nanoparticle to disappear. And so we'll talk about all of those elements in my discussion today. And if you go back to this Abraxane therapeutic, it really follows a lot of those same kinds of laws, if you think about it. And so in the case of the nanoparticle, it's made out of albumin, which is a biological molecule. Your body knows how to handle it and knows how to metabolize albumin. And so it's very non-toxic. Of course, the drug that it's carrying is very toxic, but that's by design. The drug has to be toxic to kill the tumor cells. It also follows its programming, as some of you probably know. Most nanoparticles that are used to treat cancer follow what's called the EPR effect, the enhanced permeability and retention effect. the nanoparticle has a means of selectively localizing to the tumor site. And finally, because albumin is fairly easily degraded by the body sees a lot of albumin. You have a lot of albumin in your bloodstream right now. The body knows how to degrade it and to get rid of it. So those are our 3 laws. So what I'm going to tell you guys about today in my story is how we use chemistry to build our nanostructures and how our nanostructures are used. And we're not going to be talking about albumin anymore. So we're going to think about a different type of nanoparticle. The nanoparticles that we work with in my lab are these porous silicon nanoparticles. So what you're looking at here in the picture is a silicon nanostructure made out of crystalline silicon. And it has a highly porous structure that is idealized in this cartoon here. We'll talk a little bit more about that material here in a minute. First thing you have to think about when you're putting this nanoparticle into a nanomedicine mode is it has to accommodate the drug in some form. And so we have to think about ways that we can place the drug inside that nanoparticle in an easy fashion and have it stay in there for some period of time. I'll talk about that in a few minutes. We have to seal the pores so the drug stays in there. It doesn't just leak out as soon as we put it into our syringe. It has to come out programmatically. We want it to come out ideally when it actually hits the target that we're trying to treat. And then typically then the programming comes in, and this is now again the chemistry. We have to develop various different types of chemistries on nanoparticles to make them behave in the body. They have to circulate for long periods of time, so that involves what we call stealth coatings. And we have to attach, typically in this nowadays, we attach targeting groups, so chemical molecules that have a specificity for the type of tissue that we're interested in. And so all of this is based on trying to engineer a nanostructure to do very specific functions. The disease I'm going to talk about today is going to be treating bacterial infections. And so it's maybe a sign of the times in nanotechnology that we're no longer just focused on treating cancer. The first anti-cancer nanoparticle was developed more than 30 years ago. And in fact, it's so old, it's off patent now. This is a drug called Doxil. There's a lot of money and a lot of effort that goes into developing new types of cancer treatments, whether they're small molecules or nanoparticles or whatever. And the chart shown on the right here just gives an indication of the kind of investment that's being made in drug products for treating cancer. I took this data from a relatively old study now, back about 7 years ago. There were 800 cancer therapeutics somewhere in the pipeline. And again, this could have been a nanoparticle, or it could have been a molecule or an antibody or various different types of cancer treatments, all generally. The investment in that area at our federal level in the U.S. National Institute of Health is on the order of about $20 billion a year. Or the amount that NIH puts into it is about $5 billion a year. And there's a much larger investment coming from the pharmaceutical industry as well. It's not shown here because that number was hard for me to pick out, but it's very large. And then contrast that with antibiotics. Antibiotic systems, there's at that time in 2014, there were less than 50 antibiotic products out there trying to treat bacterial diseases. On the left is shown a chart that was done by the Wellcome Trust Foundation that indicates the problem we're facing here. We're putting a lot of money and effort into treating cancer, and it's good because it's a very desperately bad disease. A lot of people die from this disease every year. But the Wellcome Trust predicted that by 2050, more people will die from antimicrobial resistant bacterial infections than from cancer. And that's because we have very few antibiotics that we can use today, and these strains naturally mutate just like cancers do. And so it's very hard to kind of keep pace with those. So that's the kind of a motivation for why we were thinking about using nanoparticles for treating bacterial infections. And again, I should point out that part of the reason for that is because now cancer therapeutics are so well accepted that nanoparticles are now being thought about as being used in other diseases. In fact, the coronavirus epidemic has proven that nanoparticles can be deployed in large quantities for large numbers of people. As you may or may not know, both the Moderna and the Pfizer vaccines are mRNA. They're a nucleic acid-based therapeutic, but it's loaded inside a nanoparticle, so-called lipid nanoparticle that I'll talk about in a few minutes. So let's talk about infections now. So this picture just shows an image from a review article just showing a mouse. It's various stages of infection in different areas. And the things that are important to keep in mind for my talk today is we'll be talking a little bit about these so-called Kaplan-Meier curves or survival curves, where we're looking at a bacterial infection in an animal, and typically how we do our readout or how we do our experiments is the animal is infected. I'll be talking about lung infections today. The animal is infected with the bacteria, and then the animal, you'll have a series of animals undergoing the experiment, and over some period of time, in this example shown here, after about 8 days, all of those animals have, or many of them have died off because the infection is so desperate. And so this is kind of the starting point. This is what would, the bad thing that's about these bacterial infections is they can be very lethal. And so what we're going to try to do is improve the percent survival shown on the y-axis there, increase the survival with our nanostructures. So let's come back to our nanoparticles here. So how are we going to do that? The first piece of that, as I mentioned, is accommodating a drug, sealing the pores, and then loading some chemistry on the surface. And so the first example I'm going to talk about today is going to take an example with a drug called vancomycin. We're going to use a sealing chemistry using calcium silicate. And the surface chemistries are going to be involving two different kinds of molecules, polyethylene glycol, which is going to help this material evade the immune system and circulate in the bloodstream for a long period of time. And then we're going to use a targeting group called a peptide. CARG is just a short form of abbreviation we use for the peptide. And that peptide has some specificity for the disease, the bacterial infection that we're going to be talking about here. Okay. So let's first talk about that material and how do we engineer the material to load the drug. And so first thing we really want to, I need to stress with you is some basic chemistry of how silicon works in water. And so it's shown in this slide is a cycle kind of going around along the top of the cycle is a silicon oxide material being dissolved with adding water. We call that process hydrolysis to make SiOH4, silicic acid. That's the bioavailable form of silicon that you have in your body right now. We eat a lot of silicon in our diets, and SiOH4 is the water-soluble form of silicon. So when that silicon oxide dissolves, it makes that byproduct, and then it goes into it basically opens up the silicon structure. And then the bottom part of this slide is important. The silicon nanostructure, if it's made out of crystalline silicon, is readily oxidized. And so the silicon nanostructure will be oxidized by even a mild oxidant such as water or a buffer solution to grow that oxide. So you grow an oxide on the silicon, the oxide dissolves, and then you grow more oxide, that oxide dissolves. And so you kind of could go continuously through this loop, and eventually when you go around this cycle enough times, all of the silicon is dissolved away. But it's two features of this process. It's both this oxidation and this hydrolysis that are important for this reaction. So I'm going to talk about that hydrolysis reaction first, because that's something that we're going to harness to load our drug. And so I have to back up for a second and talk about some very simple chemistry that is well known to most of you who've, at least you've seen it. Everybody in this on this call, I'm sure, has seen this chemistry in action. You've probably seen a cement mixer mixing up cement and spreading it out and making a building or a sidewalk or some kind of a bridge or structure. The way that cement works is it's basically sand and calcium oxide mixed together with a little bit of clay, aluminum oxide. And then the silicon dioxide and the calcium oxide react with water. So when you take that powder and you mix it with water, as you know, it kind of becomes this sort of sludge that eventually after a few hours will set up into a solid mass. And the chemistry that's doing that is actually making it precipitate. The calcium ion in the calcium oxide is dissolving in water, and the silica is also dissolving in the water and making a product of calcium silicate, which is shown at the bottom of the slide here. And that calcium silicate is a very insoluble form of calcium. Basically, that's the glue that holds concrete together, that holds cement together. So we're going to try to harness this same chemistry in our silicon nanostructures. Now, our silicon nanostructures aren't made of sand, but they do have silicon dioxide in them. And so that top reaction shown on the slide here is just the simple hydrolysis chemistry of silicon oxide. So when you put sand in water, it'll form to a very low, small amount this silicon tetrahydroxide, or silicic acid as it's called. That's on the top equation. And the second equation shows the precipitation reaction, basically the same reaction that occurs in concrete or in cement. And that is a silicon tetrahydroxide reacts with calcium ion to make calcium silicate. And so in the presence of our nanostructures, if you think about it now, remember I told you that our silicon nanostructures are dissolving continuously when they're in water. They make silicon oxide, they do the silicon hydroxide. If we add some calcium ion into the solution, we'll naturally form this, basically this cement, this calcium silicate phase within the porous nanostructure of our material. And so it's shown in the bottom part of this slide as an example where we added our silicon nanoparticles to vancomycin. And the vancomycin is an antibacterial drug that we're going to be using here in a few minutes. That vancomycin gets trapped in the pores and sealed by this calcium silicate precipitation chemistry. So you basically locally generate cement inside that very, very small nanometer scale structure. Again, these nanoparticles are really nanoparticles. They're on the order of 100 to 200 nanometers in size. So this reaction is very localized in a water solution, and it's very focused on just our silicon nanostructure. And so if we do this chemistry right, we end up with particles that have a very simple sealed drug sitting inside the nanoparticle. And I'll show some examples of that here in a few minutes. One other thing I have to point out is that the next piece of this, again, remember we talked about targeting the nanoparticles and getting them to bind selectively with a peptide. And I don't have time to talk about this. These peptides are developed by my collaborator who's shown here on the slide, Erkki Ruoslahti. He spent his career developing methods to build peptides that have some specificity for certain types of diseases. And in this case, we borrowed some peptides from him that will selectively home to bacteria in the body. So primarily the one on the bottom, Staph aureus, he did a series of screens where he found some peptides that had some affinity for Staph aureus bacteria in the body. And so we use those peptides in our experiments. And so what's idealized here in this picture in the center of your slide, that little green semicircle, it's marked peptide. That's one of his peptides. It has some specificity for Staph aureus bacteria in this case. And so this slide sort of summarizes a little bit of what's going on here in the chemistry. You can see vancomycin, the molecule shown on the left, the sealing chemistry that we did to load it with calcium silicate generates particles that are on the order of about 180 nanometers in size. And we took those nanoparticles, a picture of them is shown on the lower right. If you take those nanoparticles and disperse them into water and measure how fast that vancomycin drug comes back out, it leaks out basically over the space of about 10 hours in a buffer solution at body temperature. So now we have a nanoparticle. It's locked a drug inside that will come out. It'll slowly dissolve out. And the trick now is to have that nanoparticle circulate in the body and have those peptides attached to the disease before all the drug comes out. Because once they're attached to the disease, now they can release that drug locally and be more effective at killing off the bacteria. And so there's some data just showing the types of, the advantage of using this targeting peptide. So maybe I'd focus your thoughts or your sight just here on the bottom right-hand side of the slide, where you see a bar chart comparing the bars in red, which are the targeting peptide with our silicon nanoparticles. We idealize or abbreviate that as PSINP, so porous silicon nanoparticles. CARG, we call it CARG, is the targeting peptide. And the green bars is a control peptide. And the control peptide is just a peptide that we know does not have any specific selectivity for any tissues in the body. And this experiment was run on mice that have a lung infection with Staph aureus bacteria. And you can see that the red bar highlighted for the lung is much larger than the green bar that highlighted in the lung. And so what that says is this targeting peptide has some selectivity for getting into the lungs of infected mice. What's not shown here is we did a control experiment with mice that did not have any infection. And in there, we don't see any targeting by that peptide. And so the peptide doesn't help that particle get into the lungs any better if it's there or not there. And so here now, kind of this demonstrates that that peptide that Erkki Roslahti gave us actually does have some selectivity for getting into the lungs of infected mice. And so kind of just wrapping up that basic concept of the story then is what we found is that when we can get these nanoparticles to selectively home to the lungs, and when they're there, those particles dissolve and they release this vancomycin drug. And so the data shown on the right kind of compares that nanoparticle to just what happens if we added a free drug, just added the same amount of drug, vancomycin, the same amount of vancomycin, but just the way that a doctor normally would administer it. We just inject the patient, in this case a mouse, with the vancomycin. And you can see again these little survival curves here. So we have in the blue dots there you see if we just give free vancomycin to this animal, after about a week, 40% of the animals are still alive. So most 60% of the animals will die. So the vancomycin is effective, but just not as effective as it needs to be to save all these animals with this particular animal model. The black dots are just a control that show that basically after about 2 weeks, the animals, all but essentially one animal, have died. And then the most important trace there is the red trace that shows the targeted vancomycin nanoparticle, where if we have the targeting group and that drug located in that nanostructure, we can actually rescue all of the animals in the study. So if the animals are infected on day 0, by day 1, if we give them the injection of the nanoparticles, it will completely cure them of the disease. So that was a really nice result. We found that now we can maximize the effect of vancomycin. We can deliver the same amount, the same dose of vancomycin, and have it be more effective if we load it into a nanoparticle. Okay. So that's an interesting story. It shows that you can use a nanoparticle and target a nanoparticle to make it more effective. But the next part of my story is going to be trying to look at the same problem now, but using a different modality, a different type of therapeutic. So again, this is a picture I showed you previously. This is a mouse who's very sick, has disease everywhere. Again, we're going to focused on a lung disease. And again, this is our survival curve. Now, remember, this data that I showed you here is just kind of a notional example of a mouse getting infected with a bacteria and most of them dying away. But you'll notice in these survival curves that after day 8, there's still one little animal. There are a few animals that are still surviving. And why did those animals survive? I mean, why are they still alive after 8 days if we've given them a lethal bacterial infection? Well, the main reason is these animals have an immune system just like you and I do. And some of those animals are able to fight off the infection. And so they have a good immune system, and their immune system is a little bit better than the other mice. And so they're just able to fight off the infection and not die. And of course, the immune cells that are involved in this kind of process are lymphocytes, neutrophils, and macrophages in particular. The 3 cells in the blue that are shown there on the right are the immune systems, the cells in our body, the immune cells in our body that are designed and programmed and grown to fight off these types of infections. And so what is it about that hero mouse, that one mouse that survived for 8 days? Can we mimic that in all of our other mice? So it turns out that one of the biggest problems with this disease in the lungs is that the macrophages get into the lungs and they try to fight off the bacteria, and you get so many macrophages in the lungs and so many other immune cells flooding into the lungs that they create a massive immune response, an inflammatory response. In COVID, the COVID is a viral infection, but you've seen the same thing in patients who have a very severe case of COVID. They have what they call the cytokine storm. You may have heard that term. What that means is basically the body, its own immune system, is overreacting to that disease. And in fact, that's how most people die, and most animals in these systems, in the bacterial infection of the lungs. This is how they die. Their lungs overreact and they flood with fluid and they get massive inflammatory response. And basically, before their immune system can kick in and kill off all the bacteria, their lungs just fail. And so that's, in a sense, that's an overreaction of our body's immune system. And so what we thought we might be able to do here is just to dial down that reaction of the immune system. And so to do that, we're going to target macrophage cells. So in this experiment, we're going to make nanoparticles that have an ability to selectively go to macrophages instead of the bacteria themselves. And we're just going to tell the macrophages to slow down, to tame them a little bit and keep them from being so infective and so inflammatory. And so here's the process that we used to do that. Excuse me a minute. Okay, sorry about that. I don't know how to silence my real phone. You can still hear me okay? All right.
Yeah.
Yeah, okay. All right, so so the way that we're thinking about how we could do this was to deliver a gene and a nucleic acid, so-called siRNA. And siRNA, if it gets to the right place at the right time, has an ability to reprogram cells. And in this case, we're going to use an siRNA that is going to reprogram the macrophages and tell them to stop being so inflammatory. And this, we didn't invent this or discover this. This is known in the virology community or in the infectious disease community. The IRF5 gene is known to induce this inflammatory response and turn on the macrophages into their inflammatory phase. And so if you can silence that gene, you'll shut down that inflammatory response. So that's the therapeutic we decided to deliver. It's not going to kill bacteria. It's just going to silence or slow down the macrophages from becoming overreacting. We're going to use a different targeting peptide now. CRV is one that actually targets macrophages instead of bacteria. And we're going to coat these things with a lipid type coating to protect it. Now, lipids are important, and as you guys probably all know, the lipid nanoparticles are the nanoparticle formulations that's used in the COVID vaccines. So the Moderna and Pfizer vaccines both use lipid nanoparticles. Why do they do that? Kind of for the same reason. The lipids have an ability to protect a payload, in this case, an RNA payload. And so we kind of borrowed that same concept from the community, using a lipid coating to help us improve the ability of that therapeutic to do its action. The challenge was with the current types of technologies, including COVID vaccines, it's really difficult to load this highly negatively charged RNA into a lipid. And so an example shown in the table on the right, and generally speaking, like a lipid-based nanoparticle or a liposome or a kind of these hybrid systems. We surveyed the literature about 4 or 5 years ago, and there was no nanoparticle that could deliver or load a nucleic acid, these highly negatively charged RNA or DNA therapeutics, to a level above about 15%. So that's one of the challenges. How do you get a lot of RNA into a small nanoparticle? Because of course you can get more in there, you can get it to be more effective. And so then we kind of go back in literature and look at how people have stabilized nucleic acids and how the body and living systems stabilize nucleic acids. And it turns out that it's a kind of well-known in that community that you can actually precipitate DNA by adding calcium ion. In this case, it was calcium phosphate or calcium chloride, because calcium phosphate also makes a very insoluble compound very similar to the calcium silicate that we were talking about a few minutes ago. And so we thought, well, let's just try to use a silicon sealing chemistry that we use for our other molecule, our vancomycin, and see if the same sealing chemistry can work on a nucleic acid. And the beauty of that is that calcium ion actually has an ability to help compress and condense nucleic acids. And so this calcium ion sealing chemistry, we were actually quite surprised to find out that it has a remarkable ability to load a lot of nucleic acid into the nanoparticle. So for example, shown here in the green, the calcium ion silicate sealing chemistry that we use for siRNA gets us somewhere between 20 and 30% by mass loading. So already at least twice, or 1 to 2 times, or 1.5 to 2 times more drug loaded into the nanoparticle. And in this business, even a doubling the amount of drug that's in the nanoparticle is a big deal. And so here's kind of pictures of what these things look like once we've loaded them with nucleic acid. So on the left, you see the nanoparticles that are empty, the calcium ion sealing chemistry. You can kind of see how these porous silicon nanostructures get clogged up. The calcium chemistry tends to make them darker, denser. You don't see these nice open pores that you see in the empty particles. And they seem to also include siRNA in them. So we did this chemistry and it worked. We got a lot of RNA in there, but the challenge was it dissolved out too quickly. These particles, and you can see this in the bottom trace here, the particles gave up the nucleic acid within about, just within an hour or two, almost all of the nucleic acid had come out. And by 18 hours, basically our particles had completely dissolved. And so that dissolution rate was just way too fast for us, and it just was not going to be effective. And so that's where we borrowed this trick from the lipid nanoparticle community. And so we took these nanoparticles and we coated them with a lipid on the outside. And you can see what that does for our drug. Now, the green and the red traces there are using these liposomes or lipid coatings to protect that nanoparticle. And now when we coat that nanoparticle with this lipid, it dissolves much more slowly and it releases the RNA payload much more slowly. So this pace of release was now appropriate and pretty much a good match for how long we thought our nanoparticles would circulate in the body. And so this chemistry worked perfectly, but we had to add that lipid coating to get it to behave. So I just spend a minute or two talking about lipids and why they're important. So the lipid structures that we used are borrowed from the literature. There's a huge amount of research that's gone into lipid-based nanoparticle systems, as you guys can imagine. And it's accelerating dramatically nowadays with the advent of the coronavirus vaccines, because that was a global experiment that proved how effective liposomal nanoparticles can be. When lipids hit cell surfaces, they do one of two things. They typically do what's called endocytosis, what's shown on the right-hand side of the slide. That nanoparticle hits the cell surface and it basically gets enveloped by the cell membrane and taken up into the interior of the cell. But there are some formulations of lipids, if you do it right, you can get a formulation of lipid that when it hits the cell surface, instead of getting taken up by endocytosis, it fuses with the cell membrane, so-called fusogenic lipids. And so it basically becomes part of the membrane of the outer membrane of the cell. And so whatever payload sitting inside that lipid, and it ends up getting just dumped directly into the cytosol. I don't have time to go into it, but putting nanoparticles into endosomes is a bad idea. They tend to be very ineffective, and most people who use nanoparticles for delivery go by this endocytosis pathway. They spend a lot of time trying to figure out how to get the nanoparticles to escape the endosome. So on the left-hand side, that strategy, I think, is a much more effective one. It certainly was for us, by just avoiding the endosome entirely and going straight into the cytosol of the cell. So how these work is kind of just illustrated in this cartoon here. We're doing what we call a fusogenic liposome. Oops, sorry, this is labeled backwards. The fusogenic liposomes are on the right, the non-fusogenic liposomes are on the left. And so a fusogenic liposome fuses with the cell membrane and the non-fusogenic liposome gets taken up by endocytosis. So sorry about that, the labeling of those two processes is backwards. And so in the end, what we're trying to do is what's shown in the center. We want this nanoparticle to hit the cell surface, dump its payload directly into the cytosol. And then the beauty of this is with that protective coating gone, now our nanoparticle will dissolve very quickly and release its RNA payload. And we found, in fact, this was very effective. So the kind of basic concept is you have this targeted nanoparticle, it hits the cell surface, it fuses, it dumps its payload into the cytosol, the payload dissolves very quickly, and that nucleic acid therapeutic comes out where it can be very effective. I'll skip over some of this data considering the amount of time I have. Let's maybe go to the final slide here, showing how effective this was. And so what we found is if we were able to, again, remember this nucleic acid is designed to shut down this macrophage response, the inflammatory response of the lungs. Remember, I started this story telling you that I wanted to retrain all the mice so they all had that ability to fight off the infection on their own, using their own immune system. And we just had to slow down the macrophage response. And that's what's shown here. And so the green dots are the actual fusogenic therapeutic nanoparticle, showing that all the animals are actually rescued under this condition. If we just give a control injection of a buffer solution, then all the animals are dead within 4 days. And if we don't use a fusogenic lipid, but we use the same targeting peptide, it's effective, but not nearly as effective as using this fusogenic coating. So again, shows how important it is to avoid that endosomal uptake with nanoparticles. So kind of long story short, in the end of the day, we found that these nanoparticles can target the cells very effectively. They can shut down that inflammatory response. And when that inflammatory response was shut down, now the mice have their own immune system being able to kick in and to fight off the infection. And so at least in this model of infection, this is a Staph aureus lung infection model, we were able to completely rescue all the mice just by slowing down that macrophage overreaction. So I think that basically, just to kind of summarize what we did with that, we had to do a lot of engineering of our nanostructures. We talked at the beginning of my talk about how you do the chemistry is your computer program, if you will, or the lines of computer code you're writing. We had to develop chemistries to load our nanoparticles in very selectively and load our drug into those nanoparticles. We had to figure out how to coat those particles to get them to behave in the body and circulate through the bloodstream and target specifically certain cell types. And that was really done with this combination of these fusogenic lipids and the targeting peptide. So kind of just to sort of summarize everything that you've talked about today, I spent a little bit of time talking about this self-sealing, what we call the self-sealing porous silicon nanoparticles. You put these nanoparticles into solution, you squirt in some calcium ion, and they naturally just trap anything that happens to be around inside their structure. They dissolve in a very non-toxic fashion. We looked at a lot of cell toxicity and animal toxicity studies were performed to get to this stage in our studies and found they're very, very low toxicity. And that they're really good at targeting. So we can have them target specific locations. I showed you 2 examples today of just directly attacking the bacteria and targeting them. And then another peptide that was used to directly target macrophages that would then basically shut down an inflammatory response. With the case of the bacterial targeting, we showed that we could deliver vancomycin much more effectively, so we didn't have to use as large of a dose to be able to get the same effect. And then with this fusogenic nanoparticles was really an enabling feature of getting nucleic acid therapeutics into the cell and getting to where they need to go to do their business and to shut down this inflammatory response in a macrophage lung infection model. So with that, I'll thank you guys for listening and for your attention, and I'm happy to answer any questions you might have. The slide here just acknowledges my collaborators. I should really highlight Erkki Ruoslahti, who I mentioned already, who developed the targeting peptides for us. Sangeeta Bhatia at MIT, who did a lot of the really hard work and helping us develop these nanoparticles for these bacterial infection models. Jinyoung Kang developed the calcium silicate sealing chemistry in my lab, and BJ Kim, the first two authors listed there, focused on this bacterial treatment model. BJ developed the fusogenic lipid coating that was so effective at delivering siRNA to cells. And with that, I'll take any questions you might have.
Yeah, great. Thank you. Thank you very much, Mike.
Thank you.
It was a great talk, and yeah, and very encouraging, I will say, with different things that you presented. I want just to invite all the people that is following us in YouTube. There are some people there. Now you can simply write down your questions now that we are on, that we are live now. No, yeah, simply right there. I just wanted to start with the questions, Mike. Considering this part of the cement, it was very, very interesting. First, for the reaction, you are considering that this silicon dioxide is converting to silicon hydroxide. Does it occur at certain pH faster?
Yeah. No, a good question. So those of us who've done synthetic chemistry for a living years ago will know that if you ever take your glassware and you put it into a strong base bath, it'll eventually all dissolve away. So glass does dissolve in strongly caustic solutions very quickly. Of course, at neutral pH, your windows don't dissolve when it rains. And so we know that, and also in acidic conditions, glass is quite stable. But that's really in the bulk form. In the nano form, you've got a lot more surface area there. And even when it rains on your windows, you have, you lose a few, a nanometer or so of silica every time it rains. It's just that you don't notice that because it's a very small amount. But silica will dissolve in water. It's just fairly low solubility. And so that's really what you harness. So for example, in your bloodstream, you have about 5 parts per million of silica, dissolved silica. And so, that's because we live on a planet that has a lot of silicon in it. And so you ingest a lot of silicon in your diet daily. So the silicon is very pH dependent for its dissolution process. It's also very dependent on nucleophiles. So even if you're at neutral pH, if there's some amine buffers in there, nitrogen bases or phosphorus bases, they tend to dissolve faster. Ions also tend to help the dissolution process. So magnesium and calcium tend to suppress the reaction, because you make that magnesium silicate or calcium silicate. But yeah, so that's one of the challenges in these systems is you want to design the surface area, the porosity of the material, so that it will dissolve at a rate that's not so fast that you overload it reprecipitates somewhere else in the body, but it dissolves and then gets excreted.
Yeah, that's nice to hear. We work also on some silicon microstructuring stuff, or porosification and stuff. And yeah, it is very encouraging, I would say. And that stuff of the calcium, actually, we are, as you know, fabricating some batteries, and the last time we have been working on calcium oxide batteries.
Oh, okay. Yeah, so that chemistry is important in that domain as well. And it's interesting because in the battery community, your chemistry challenges are kind of opposite. And in our world, we're trying to get these materials to dissolve ultimately. We don't want them to be stable. In your world, they have to be really stable. So it's a totally different set of criteria for what kind of chemistries you can choose.
Yeah, it was very interesting that formation of
looks like
cement.
We have a question in the chat.
Yeah, we have a question there. Yeah, if this research will be applied also for water decontamination.
Yeah, it's a really good question. We haven't looked at that. of course, silica is used as a clarifying agent in many processes. They add silica beads to beer, alcohol, beer you drink. They clarify it. So a lot of the beers that we drink, well, Enrique, I know you came from Germany, spent a lot of time in Germany. The Germans don't believe in clarifying their beer, and so they don't do it. But in Mexico and the US, we tend to filter our beer through silica, and that does take out, particles and things. The specific chemistry that we're doing, I don't know if it has anything really to say about decontaminating water. The things we're really trying to get rid of there would be like heavy metal ions, I guess is what you might be asking, Miguel. There we haven't done a lot of work, and I'm not sure there's much that the silicon oxide system lends to heavy metal decontamination. It's not my area of expertise, sorry.
Also, now that we are waiting for more questions from the audience, I wanted to simply ask you some comments about this multidisciplinary work that you are performing. Actually, you listed many people there, and I think are from different areas also, as you presented also some people from the lipid stuff, no, and some organic chemistry and stuff like that. I don't know if you could comment a little bit about that.
Well, I think, that's a really good point, Enrique. I think, we've both been more or less trained as chemists, and I'm not sure what our audience's background is here. But that's the beauty, I think, of the material science community and the people who work in material science, is we have to have a large range of backgrounds. I mean, I really do not have expertise in bacterial infections per se. I have collaborators who do, and that's the really important thing. And materials science in many ways brings the world together, rather than it forces you. If you want to be successful, you have to work with other people who have expertise in their domain. This is true in the biomedicine in a huge way. You have to have people who know the biology, you have people who know the chemistry, you have people who have to know the materials, and materials properties are all really important features. So yeah, I think that's the reason I love being a materials scientist and being a materials chemist, is you get to talk to lots of different people with different perspectives. So yeah, we're a very inclusive community, you guys. If you're any undergraduates or people thinking about going into this field, I think it's a really great domain to work in.
Yeah, actually, there are many people from the audience now that are from the electronics part, that some people that are from, for example, as my wife do, this signal processing, for example, and people also for some sensors. And yeah, I think we all can communicate and do some stuff together.
Yeah. And the electronics community is also heavily involved in materials, obviously. And that's another example I didn't bring up. But yeah, besides talking to biologists, if you're working in the more of an electronic domain like you do in batteries or something, you really have to be engaging with people that know circuits and the other impedance and other issues that we don't get trained on as chemists, right?
Yeah.
Necessarily.
But yeah, we can communicate very, very nicely, and it's also what I like from material science and to know people like you there. Actually, I must mention that you are having a school on borosilicon every summer. If anyone is interested from the audience, and for different applications, I think, no, Mike?
Yeah, I could give a plug for that if I could get this into the chat. Let me see if it'll work. So yeah, we run a summer school every year. I run a center now that is involved in giving training on nanomaterials and nanomedicine and in another number of different domains. So I'm not sure who I sent this to. It might have been to you, or it might have been the whole community. I haven't used this one. Did that come to you? Did that go to the whole group?
Yeah. Now,
okay, great.
Now
okay.
Very good.
And so that's our website. If you're interested in coming, I had actually this. I think I was telling Enrique earlier before we started the recording. I had three students from Mexico this summer in the lab. One from Portugal, one from Korea, a couple from Korea, a few from China, and so it's a very international group. A lot of them are undergraduate students. A lot of them are master's students, and some PhD students, some postdocs. We even have visiting scholars who come. So Enrique, you want to come and do the summer school? You could be a special trainer. We could also participate. We'd love to have you. Yeah, and there's the IEEE runs a summer program as well. So I put that into the chat. And so, all of these are meant to try to get people engaged in research, give you an experience in a certain domain, a certain area, in our case, sort of nanomedicine or nanomaterials used to apply to medical or other chemical kinds of problems. And so if that's something that's interesting to you, and trust me, even if you spent your whole life doing circuits and you only know how a transistor works and that's all, we can still use you, because as long as you're eager to learn and interested in learning something a little bit outside of your comfort area, we'd love to have you apply.
Yeah, exactly. And yeah, actually, I was discussing with some of my students. Many of them are already are now there watching this transmission, and I think they are a little bit shy to ask questions. I don't know.
Oh, no problem. Oh, well, you've got my website there. You're welcome to have anybody follow up with questions. My email address is in there as well, and I'm happy to follow up with any questions that people might have.
Yeah, I was telling the students, look to the CV of Mike Saylor. No, I want to be like him in 20 years or so. So active, as you are. Yeah.
Okay.
Nice. Yeah, I'm very happy to have you here, and I hope we will meet us again in the next meeting.
Yeah, once we can travel again and go see each other, I'm looking forward to it.
Yeah. So I think we can close here, and thank you so much. It was a pleasure, Mike.
Okay, thanks very much.
We'll see us in the future.
Okay.
Bye.
Bye.